Welcome to the most impactful week in the movement to end prostate cancer. We're so glad you're here. This event is about you and connecting with experts, advocates, and communities from across the country. Together, we are Zero Strong. Please join us right now for the virtual Zero Prostate Cancer Summit.
Hi, and welcome to Zero's 2021 Virtual Summit. I'm Shelby Monier, Zero's Vice President of Patient Programs and Education. It's my pleasure to introduce you to Dr. Nima Sharifi, who is joining us today to talk about androgen, hormonal therapy, and response in prostate cancer. Dr. Sharifi is the director of the Genitourinary Malignancies Research Center at the Cleveland Clinic. He also co-leads the Genitourinary Malignancies program at Case Comprehensive Cancer Center. Dr. Sharifi, thank you for being a part of our virtual summit.
I'm going to tell you about essentially some background on and work on androgens, hormonal therapy, and response in prostate cancer. So what I typically like to do is basically just start with the conclusion so you know exactly where I'm going with this and you don't have to guess. So the takeaways here are that prostate cancer responsiveness to hormonal therapy is effectively determined in part by an aspect of inherited genetics. This aspect of genetics regulates a variation in normal hormonal or androgen physiology. And we're still learning how this information will be useful in the clinic, but we're not quite there yet. So here's the outline. I'll start with a bit of background on androgen physiology. And then I'll get into how some specific hormonal therapies work. And then I'll talk about a recent discovery of the genetics of androgen physiology and how this relates to prostate cancer, as well as some other diseases as well. So I'd like to just start with some definitions so that everybody's on the same page. Genotype is a term that refers to the genetics of an individual or their DNA. And it's genotype or genetics that can in turn determine phenotype, which means the characteristics of an individual. Germline genetics refers to DNA or genetics that's inherited from one's parents. And that's in contrast to somatic genetics, which typically refers to changes that arise from a single cell. So, you know, one of the instances here is, for example, in somatic tumor genetics, where you may have a group of cells, one of which may get a mutation or a change in the DNA. And if that offers a proliferative advantage to that cell, then cells that have that mutation specifically can populate, for example, a certain area. So this is distinct from inherited or germline genetics. Other definitions, steroid refers to hormones that have this four-ring structure. And there are specific examples of steroids or steroid hormones. Some of these include androgens, or testosterone is one of the androgens. There are also estrogens or estradiol, for example, that comes from the ovaries of females. There are also glucocorticoids, which are steroid hormones. So some of the examples here are cortisol, which is one that your body makes on its own. It's known as a stress hormone. And there's also prednisone, which is a glucocorticoid or a drug that we sometimes give to patients for treatment of inflammatory diseases and other diseases. Okay, so these are all steroids or steroid hormones. Sex steroids specifically refers to androgens and estrogens. So here's our traditional view of sex steroids, in that the gonadal source is the major source, so that men make testicular testosterone, women make ovarian estrogen or estradiol, and these gonadal sex steroids have clear clinical phenotypes, right? So some of this includes sexual differentiation. So we know that androgens like testosterone are required for male sexual differentiation in development. These sex steroids can drive the proliferation of certain cancers. For example, androgens or testosterone can drive the proliferation of prostate cancer, as breast cancer, estrogens can drive the proliferation of breast cancer. And we now recognize that there are also influences of these sex steroids on immune function. So I think a lot of this has been discussed recently, for example, in COVID. So we know that males and females respond differently to COVID, and this may have to do with the strength of immune function and immune regulation. And there are other things that are performed by these sex steroids as well, metabolic function, other things that I won't get into. So those are gonadal sex steroids, so testicular or ovarian. But there are also these extragonadal or non-testicular, non-ovarian sex steroids and their phenotypes. The extragonadal sex steroids primarily come from the human adrenal gland. So the adrenal sits on top of the kidney, and it makes this hormone called DHEA and DHEA sulfate. So this steroid hormone goes out into blood, goes out into circulation, and in various peripheral tissues, for example, including the prostate and prostate cancer, can be converted to androgens like testosterone. And then depending on the tissue, so you actually have to make androgens in order to make estrogens. So the question is, like the gonadal sex steroids in the prior slide, what are the phenotypes? So what do these extragonadal sex steroids do? So this is really quite a murky area, to be frank. And our developing perspective is that this has to be viewed through a lens of genetics and metabolism. And when we do that, when we look at these phenotypes, meaning the clinical characteristics that are driven by these extragonadal sex steroids, we have to do this through a lens of genetics and metabolism. And when we do that, what they do, the phenotypes conferred, can become pretty clear. So here's a bit more background on androgen physiology. A lot of this really starts at the level of the pituitary. So the pituitary, which sits right below the brain, is needed for gonadal testosterone secretion and function. So this secretes a hormone, luteinizing hormone, that goes to the gonads or the testes, which allows for gonadal testosterone release into blood. And the testosterone, once it hits the prostate, is converted to a more potent androgen, which is DHT or dihydrotestosterone. And that enzyme is 5-alpha reductase that converts it over. So as I mentioned, it's not just the gonadal or testicular androgens, but the adrenals also make extratesticular or extragonadal androgens. Now, let me just say a little bit more about these adrenal androgens. These adrenal androgens are usually not directly active. This DHEA and DHEA sulfate that I mentioned previously is the most abundant one, so the one that typically has the highest concentration in blood. And we oftentimes refer to these as weak androgens, which really typically have to be converted to strong androgens to perform their androgenic functions. So here's another sort of slide depicting what androgens and androgen receptor do. So again, gonadal testosterone goes into circulation, and in the prostate and also in other tissues, it's converted by 5-alpha reductase to this more powerful androgen, 5-alpha dihydrotestosterone, or DHT for short. So this represents what's happening in the prostate cell and the prostate cancer cell as well. And this is the partition between the nucleus or the core of the cell and the cytoplasm, which is the outer part of the cell. The androgen receptor, typically when it's not active, is not in the nucleus. And when DHT binds the androgen receptor goes into the nucleus, which is where the DNA resides. And this activates a number of androgen responsive genes. So that's what the androgen receptor does. It activates certain androgen responsive genes. This is what the androgen receptor, I'm just going to tell you a little bit about the function of the androgen receptor in terms of the parts, at least two parts of it. So if you if you elongate the protein of the androgen receptor, one of the relevant sites for function is the LBD, or the ligand binding domain. This is where testosterone binds. And this other hormone, the more powerful androgen, DHT, is even more active. So what this is, this ligand binding domain, is basically the brakes on the androgen receptor. So we know this because if you chop off this ligand binding domain, the rest of the androgen receptor is on, and it stays on. And so the brakes are, so the default is for the brakes to be on. And when the androgens bind the ligand binding domain, that's effectively cutting the lines to the brakes, which is, or pulling back on the brakes. So in total, that's how the androgens activate the androgen receptor. The other part of the androgen receptor that's important to know about is the DNA binding domain, or the DBD, which sits here in the middle. And this is the specific part of the protein which targets the androgen receptor to certain genes in the human genome, and it's required for gene activation. So one of the genes, for example, that's activated by the androgen receptor is PSA, or prostate-specific antigen, but there are many others as well. So how do hormonal therapies work? And at the risk of telling you a lot of information that you already know, I'll just sort of go through this again just to make sure that everybody's on the same page. So one of the ways in which some of these hormonal therapies work is to block testicular testosterone release. So this could be surgical castration, it could be medical castration, these GnRH acting drugs that at the level of the pituitary block testosterone release. There are also drugs that block adrenal androgen synthesis and release. One of these is called abiraterone. There's an older drug called ketoconazole, which is a bit weaker, and we don't typically use it much anymore in the US. And then there are also other drugs that block the androgen receptor access to the ligand binding domain. These are drugs like enzalutamide, apalutamide, and darolutamide. So there are various ways in which these hormonal therapies can be directed. This is the work, Nobel Prize winning work of Huggins and Hodges, who back about 70, 80 years ago, first showed the therapeutic effect of gonadal testosterone deprivation, which gives a response frequently for upfront treatment of prostate cancer. Now, this is a simplified diagram of the various biologic states of prostate cancer. And I'll emphasize again that it is somewhat simplified, but it gets a point across. And this has nothing to do with localized or metastatic disease. This here represents the androgen receptor, or AR, and it being on. So transcriptional activation has to do with it activating its androgen responsive genes. So in the setting of someone who has new, untreated, advanced prostate cancer, the androgen receptor is typically on, and it's oftentimes driven in large part by gonadal testosterone, which stimulates the androgen receptor, and you see an increase in the tumor burden. So for a long time, the standard of care for metastatic disease had been gonadal testosterone deprivation, right? So this deprives AR of its stimulus. This decreases AR activity, transcriptional activity, and decreases tumor burden. So that's a clinical response. And oftentimes after variable period of time, the tumor can start to progress again. And this occurs by androgen receptor turning back on. And what's really important here is that in this context, so these are men treated with medical castration where the androgen receptors turn back on. If you look inside these tumors, you find that the level of androgens is now elevated again. And this is because these tumors make their own androgens from non-testicular or non-gonadal precursor steroids. When they make the androgens, this stimulates androgen receptor and can promote tumor progression. So this is the resistant form of prostate cancer that we it has various terms: castration resistant, hormone refractory, etc. So, and I'll say that this is somewhat simplified because this also occurs in the context of other somatic genetic changes that also take place in the meantime, but I'm not going to get into all those details right now because it's not relevant. So how do these tumors make these powerful androgens? Well, it comes in large part from these adrenal precursor steroids. As I mentioned, DHEA sulfate is the most abundant in circulation. This is the most abundant steroid hormone that men and women have in blood. And this can be converted over to this powerful androgen, DHT, in the prostate tissue itself. So one of the questions becomes, are these androgens made by the prostate cancer required for tumor progression, or are they basically a bystander? Well, we have a series of large practice-changing clinical trials which show, for example, here with this drug abiraterone compared with placebo. So this blocks the synthesis of adrenal androgens, and it increases survival in the setting of disease that's resistant to medical castration. And when we use this drug along with upfront gonadal testosterone deprivation, it actually prolongs survival even to a greater extent. And when we use these androgen receptor blockers, the direct androgen receptor blockers, drugs like enzalutamide and apalutamide, you get a similar prolongation of overall survival, right? So incredible clinical benefit. Not everybody benefits, but it occurs commonly. So this, I'd say, altogether is really high-level clinical evidence that the androgens made by the tumors are certainly required for progression. Then the question becomes, how do these tumors make these powerful androgens like DHT? So I'm going to tell you a bit about steroid synthesis or metabolism that the tumors do to make these powerful androgens, testosterone and DHT. And I'm going to draw an analogy here. So DHEA or DHEA sulfate is in many ways like crude oil, which oftentimes isn't used on its own, but that crude oil can be refined in a stepwise fashion to refined oil that you can then, put in your car and use it in various forms. So similarly, this DHEA can be converted in a stepwise fashion to this refined form in various tissues, including prostate cancer, to more powerful androgens. The very first step in this refining or metabolic process occurs by this enzyme or protein called 3-beta HSD1. And so this is a regulator, okay, of converting the crude to the refined oil or androgens. And this is the stuff that effectively can stimulate androgen receptor, which can promote tumor progression. So this enzyme 3-beta HSD1 comes from or is made by this gene called HSD3B1. And a couple things that are important here is that this enzymatic activity is required for all pathways to make these potent androgens from adrenal precursors, etc. There's a very common germline variant in this gene that encodes for a difference in the protein, and the frequency of this germline variant really differs by race and geography. And there are two forms that I'd like to tell you about. One that we term the adrenal restrictive genotype, and one that we term the adrenal permissive genotype. So the adrenal restrictive genotype is just a single change in the DNA. 1245 position is A. And basically what this does is it encodes for a protein or an enzyme that's rapidly degraded in the cell. So you have lower levels of the protein or refining machinery. And this basically enables for very slow conversion from DHEA to powerful androgens. And so it leads to low levels of DHT synthesis. And this is why we call it adrenal restrictive. It's restrictive because it limits the conversion from adrenal DHEA to powerful androgens. And in contrast, we have this adrenal permissive genotype. That same nucleotide, instead of an A, it's a C, it's 1245C. Now, in terms of the function, it encodes for a protein that's stable. So you have higher levels at baseline in the cell. And since you have more of the protein, more of the refining machinery, this enables more quick or faster conversion from DHEA to DHT. You get higher levels of DHT in the tissues. And this is why we call it adrenal permissive. It's permissive because it enables the conversion from the crude androgens to the more refined stuff, right? So what does that discovery in the laboratory look like? I'm just going to share with you a little bit of that. So this was an observation that we first made actually in the lab about 10 years ago now. And what we're doing is we're looking at how two different human-derived prostate cancer cell line models handle these adrenal androgens. So we're basically taking the cells, we're adding this adrenal DHEA, and what you see is effectively you have two totally different behaviors in these models. So you have fast conversion versus slow conversion. So what happens here, we put on the DHEA, and you can see that it's converted, it's depleted rapidly. This first step is that enzyme 3-beta-HSD1. So the crude oil is depleted, and then you get sequential conversion to this refined stuff, the DHT. And in contrast with this other model, the crude oil just sits there, and that's because this machinery, enzyme machinery, is very slow or inactive, and you get very little downstream synthesis or the refined androgen, okay? So slow versus fast, fast versus slow, adrenal permissive versus adrenal restrictive. And this is all because you get that single change in the DNA confers for a different type of the protein. This adrenal permissive or faster enzyme is in the germline or inherited DNA at a really high allele frequency, so 20 to 30%. So what's also really important is that there are several studies that have been done looking at the genomes of various populations around the world. One of them is called the 1,000 Genomes Project. And when we sort of dig into that, we find that the frequency of this adrenal permissive allele varies tremendously by race and geography. So the highest frequency of this fast enzyme, the adrenal permissive allele, is actually in Europe. And there's a north-south gradient, so that the highest frequency is actually in Southern Europe, in Italy and Spain, where it's around 35 or 40% allele frequency. The frequency of that fast enzyme is much lower in East Asia. It's around 5 to 10%. And then it's also very low in populations in Africa, typically. Also similarly, less than 10%, 5 to 10%. So when we made that observation about how in different contexts you have the adrenal permissive versus adrenal restrictive, the next question that we wondered about was whether this drives the variability in the duration of clinical response to medical castration in patients, in men treated with advanced prostate cancer. So what we suspected is that men who inherit 0 versus 1 versus 2 copies of the adrenal permissive allele, when treated with medical castration, may have progressively worse outcomes, because when you inherit more copies of this more active enzyme, this will allow the tumors to make their own androgens more quickly, and that would enable resistance to occur more rapidly. So we retrospectively looked at almost 120 patients who were treated with medical castration after their PSA came back after local therapy. And what we found was that men who do not inherit this allele, the FAST allele, have the best outcomes. Those who inherit one copy of this adrenal permissive allele, or FAST enzyme, have outcomes that are a bit worse. And those who inherit two copies of this adrenal permissive allele actually have outcomes that are even worse compared with the 0 or 1 copy of the adrenal permissive allele. And then what we did is we collaborated with colleagues at Mayo Clinic to see if what we found here in Cleveland also applies in other men that have already been treated in the past there. And we find indeed that that is the case. So again, in the green are those men who do not inherit the adrenal permissive allele. Blue is they inherit one. Red is those who inherit two copies of the adrenal permissive allele. Again, this is all after medical castration. And then shortly after that, the group at University of Utah published an independent validation study. And again, in the blue, you see those who inherit two copies of the adrenal permissive allele have significantly worse outcomes compared with everybody else. And now there are at least 8 clinical studies that have been published here and described, showing that this genotype or this aspect of genetics with a single nucleotide confers different outcomes in men treated with medical castration. So this is what we think is happening. Again, that the adrenal restrictive allele effectively acts as a dam to hold back the adrenal precursors, the DHEA. But when you have inheritance of the adrenal permissive allele, this effectively opens up the floodgates and allows for conversion to more powerful androgens that allow for more rapid progression. So these are the various studies. This first one was our study looking at 3 different cohorts from Cleveland and Mayo Clinic. And then the one in Utah, we also looked at this with our colleagues from Boston, Dana-Farber, effectively showing the same thing. And since then, other groups from around the world in Japan and Spain have really validated that what we've observed in the US also applies to other populations that have been studied around the world. And we've also subsequently looked at this in a multi-institutional phase 3 study that basically showing the same thing. So now I think it's probably fair to say that we have a really firm handle that links this aspect of genetics, meaning inheritance of adrenal permissive versus adrenal restrictive allele, that links that genetics to a certain aspect of biochemistry, meaning how cells specifically handle steroids or metabolize steroids in converting inactive adrenal precursors to potent androgens. So it links not just those two, but something to a clinical phenotype, meaningful clinical outcomes in men with prostate cancer, right? So these 3 things are linked together, I'd say, quite tightly. And at the same time, unlike, the gonadal sex steroids, the other steroids that we know have certain functions, we know that cortisol performs a function for modifying stress. There are other steroid hormones that regulate blood pressure and salt homeostasis. We don't really know, we don't really understand what this adrenal DHEA does. And we suspected that this understanding that we've developed by studying prostate cancer would allow us to approach this question genetically, because now we've identified a major regulator that converts DHEA to downstream potent androgens, which I think links it to the phenotypes. What happens in people who inherit this different aspect of the genetics, right? So we decided to look at that as well. And this sounds like a little bit of a pivot, but this is a follow-on of the prostate cancer genetics and metabolism that I just shared with you. Now this next part, and I'll be brief about this, is a really collaborative study with 3 pulmonary physician scientists, Dr. Ben Gasson, Dr. Serpil Erzurum, Dr. Joe Zane. They're all asthma specialists. And it all really started about 4 years ago when I was giving a seminar on this aspect of HST3B1, and Dr. Gasson was in the audience, and he said, you know, Nima, this is really interesting because in asthma, we have this observation that high levels of DHEA sulfate really correlates with better clinical outcomes in asthmatics. So how could that be? Does that really link it to some way that DHEA is functioning? Is it just correlative? And the way it could work, we sort of discussed back then, is that we know that androgens have a function in the immune system, right? So again, we know that men and women have different types of immune function, immune regulation. Women in general have more severe and more frequent inflammatory diseases, autoimmune diseases. Their immune systems in general tend to be more active. And then on the other hand, when you look at certain outcomes for certain infectious disease processes, COVID-19 may be one of them. There's a lot of literature on this, that women may have some higher level of resistance. Their immune systems tend to be more active. And we also know that androgens can be immune suppressive. So this effect of sex steroids also applies, we believe, to asthma and differences in asthma outcomes for men and women. So for example, after puberty, we know that asthma in general tends to get better, milder in males and men, and may get worse in women. And that may be due to androgens. That's at least one possibility. So we suspected that this high DHEA, if it's functioning through this way of potent androgens, then it would require us to really look at this through the genetics of HST3B1. So we decided to look at that. And we initially did that in a cohort of over 300 asthmatic patients. And again, this is a highly collaborative study with 12 institutions around the country. And this is what we're looking at. So the y-axis here is a measure of lung function in asthmatics. So the higher the number, the better. The lower it is, the worse the lung function. This is something called FEV1. It's basically how much you can exhale in 1 second. And then on the x-axis here is the number of adrenal permissive or more active HST3B1 genes. So 0 versus 1 versus 2. So as you move to the right, it becomes more active. And we're looking at two sets of patients. Number one is those who are not taking oral glucocorticoids, so drugs like prednisone by mouth, and those who are. And when we look at those who are not on oral glucocorticoids, we find that there's really no association between this aspect of genetics, HST3B1, and lung function. Okay, there's no association. It's basically a flat line. And when we look at patients who are taking oral glucocorticoids, typically prednisone, we find that the lung function is highly associated with HST3B1, such that the more copies of the more active enzyme is inherited, the better the lung function. Okay, so what's going on here? What is this really all about? Well, we've really used glucocorticoids in medicine for many decades, since they were first identified, described, synthesized in the 1940s and 1950s. So it's used for a lot of inflammatory diseases. But there's this entity of glucocorticoid resistance, or not responding to glucocorticoids, that's a common problem. And I should say that, we recognize that, there are several different hormones made by the human adrenal gland. Among them are cortisol, or the stress hormone, and DHEA and DHEA sulfate. And when you give someone oral glucocorticoids or by IV, one of the problems or one of the adverse effects is that you suppress the production of the cortisol that's made by the body itself. And this can become clear or evident when you give really high doses of glucocorticoids. Okay, so if you've ever taken prednisone or higher doses of prednisone, when you come off of it, your physician will oftentimes taper it down step by step. And that's because if you take it away too quickly, your body's not making enough of this stuff on its own, and that can cause problems. So we recognize that in medicine. But what also happens that's not widely recognized is that these oral glucocorticoids can also suppress adrenal DHEA production. And we don't talk about it, we don't recognize it because it doesn't have any real known adverse consequences. But that might not be the case. So this is what we envision may be occurring in the context of asthma. So when you take prednisone, right? So what happens in these asthmatics, you take prednisone and the DHEAS drops by about 70%. So you're only making 30% of what's normally there. When you inherit the weaker enzyme, okay, the limited amount of DHEA will not allow for much testosterone production locally, and that can block the anti-inflammatory effect that you want with glucocorticoids. On the other hand, when you inherit 2 copies of the more active enzyme, even though you've limited the DHEA, this can still make enough of the downstream androgens we suspect to maintain an anti-inflammatory response. So this is the model that we're working on based on the data that I've shown you. Now, is this also validated in another cohort? So we actually looked in a second cohort of asthmatics. Again, here's HSC3B1, this aspect of genetics that we know is important for prostate cancer, 0 versus 1 versus 2 alleles, and lung function here. Again, patients who are taking oral glucocorticoids with severe asthma, the lung function appears to be highly associated with HSD3B1 genetics. So I'm just going to go to the conclusions here. So one is that sex steroids, meaning androgens and estrogens, also come from non-gonadal sources in normal physiology. HSD3B1 genetics really regulates extragenetal androgen synthesis. Not the amount of DHA that's there in blood, but how that DHA is used in tissues, including, for example, the prostate or prostate cancer. HSD3B1 dictates prostate cancer clinical outcomes in men who are treated with gonadal testosterone deprivation or medical castration. And we have very active research underway to figure out how this information will be useful for clinical decision-making in prostate cancer. So we're not quite sure how to use this information yet. What we need to do is to interrogate this aspect of genetics and the practice-changing clinical trials that have already been done to figure out how this can be utilized, for example, as a biomarker. But I do want to emphasize that it's not absolutely mature at this time, and that we still have work to do. But the same aspect of genetics, interestingly, also regulates the anti-inflammatory response of glucocorticoid therapy and asthma. And this is, who would have thought that studying a very sort of basic, fundamental metabolic molecular process in prostate cancer would give us clues, not just to prostate cancer, but to other diseases that, at least on the face of it, seems like it's not at all related to prostate cancer. But it certainly seems to have done that, at least for this story. And we suspect that this aspect of genetics, because it regulates this basic mechanism of physiology, may also regulate other clinical diseases and other phenotypes that we have yet to identify or discover. So I should say that as the leader of our research program and GE Malignancies Research Center, I see my job mainly as a cheerleader. I'm not at the bench, and there are a lot of deserving people who are very talented group who work very hard day and night to make this work move forward and to mature. And we all have a singular goal in mind, which is to make the lives of patients better, to improve what we can do in the clinic, and to try to overcome some of the limitations of therapy that we have in the clinic right now. These are some of the former members of our group who've gone on to do other fantastic things. We have a number of collaborators here in Cleveland who are really incredible and supportive, fun to work with, and again, have a common goal. I want to specifically mention Eric Klein, who's a leader of the Urologic and Kidney Institute here at Cleveland Clinic. There are many other collaborators around the country and around the world that we've worked with as well to make much of this happen. And I, specifically want to give great thanks to patients and their families. Everything that we do, even though a lot of it's sort of basic and mechanistic, we always have an eye toward the clinic. We want to figure out how the disease is occurring in patients, in men, and we want to be able to overcome the limitations that we have to develop better therapies, to make lives better. So this is all attributable to patients volunteering for research and their families also being devoted to that, because it takes effort on the parts of the families as well. And I also want to thank our funding sources: Prostate Cancer Foundation, the National Cancer Institute, American Cancer Society, the Harrington Discovery Institute, the Kendrick family, and others. So this is where we live in the Lerner Research Institute at Cleveland Clinic. This is our Cancer Institute and our Urologic Institute, and this is a little bit of a dated photo of our research group on one of our outings prior to COVID. And I want to thank you for your attention.
Thank you, Dr. Sharifi. This is such an important topic for men affected by prostate cancer to understand as a treatment option. And on behalf of the entire prostate cancer community, I want to thank you for being here and being involved in Zero's 2021 Virtual Summit.
The issues you care about most are right here at the virtual Zero Prostate Cancer Summit. Please check out the other topics, sessions, and virtual get-togethers in your Summit app. Remember, you are not alone. Join the Zero community to gain support and more information on prostate cancer at zerocancer.org.